Hello again. So now we get to the second half of the first chapter of the second section of the course. Okay, that's just too confusing. Um, uh, let's just say that this continuation of chapter 5 is the part where I'll talk about LTP1, LTP2, and LTP3. So I'm showing my modified diagram again on this opening slide. We're going to see that LTP1 seems to activate only the post-translational modifications of proteins that I spent much of the first 4 chapters talking about. And I'm going to remind you again that LTP2 adds in new protein synthesis that doesn't actually require genomic signaling. And LTP3 adds in genomic signaling. I'm also going to describe a very exciting experiment that characterized morphological changes in spines. I'm busting to tell you about that experiment, but I'm going to hold off for a bit, and we'll start by examining the also very exciting LTP1, 2, and 3. Oh, and I think this lecture will be a little short because I covered a lot in the first lecture for chapter 5. But I will post up the lecture for chapter 6 soon so that you can keep moving forward. So let's explore LTP1, LTP2, and LTP3. This is actually a model of 3 hippocampal stimulation protocols that really have told us a lot about how potentiation is mediated in hippocampal spines. This model makes use of theta burst stimulation that I've previously described. You know, the pattern stimulation that's illustrated by the red hash marks, again on this slide. There may be other slightly different versions of this pattern stimulation, but the fundamental form of it consists of brief stimulation with 100 hertz, or 100 cycles per second, of depolarizing stimulation. Then there's a brief pause, and then another brief train of high-frequency stimulation. Now, Raymond and Redman have done a lot of work with this theta burst form of stimulation. And they discovered that the magnitude and duration of CA1 pyramidal cells' responses to the stimulation differ depending upon the number of trains of stimulation administered. They examined the response to 1 train of theta burst stimulation, and quite sensibly, again, if not imaginatively, called this LTP1. Then they explored the effects of 4 trains of stimulation and called it, can you guess? Yeah, LTP2. Then they examined the response to 8 trains of stimulation, and you got it, LTP3. They found that the magnitude of the LTP was increased with more trains of stimulation, shown in the graph, kind of in the middle right of the slide. They also found the decay rates of the LTP were decreased with more trains of stimulation, shown again on that graph, but also in the bar graph at the bottom. The graph at the bottom of the slide is the time constant, or depicts the time constant of the decay of potentiation for the LTP1, LTP2, and LTP3 paradigms. The larger the time constant is, the longer is the duration of the potentiation. So you can see that the duration of LTP is greatest when 8 trains of stimulation are administered. All right, so, well, so far, there's nothing much to get excited about. But Raymond and Redman continue to explore the mechanisms underlying these effects and found differing roles of calcium signaling for each of these experimental manipulations. And that's what we're going to explore next. We'll start with LTP1 because, well, that makes good sense. So when Raymond and Redman applied one train of theta burst stimulation to Schaefer collateral inputs to stratum radiatum of CA1 pyramidal cells, they found a rapidly decaying form of LTP, shown in the open circles in the graph at the left of this slide. And then, when they blocked ryanodine receptors with ryanodine, shown in the closed circles, they impaired this form of LTP, decreasing both the magnitude, measured as the area under the curve, and the duration of the potentiation. So you're probably wondering, what the heck are ryanodine receptors? Let's talk about them for just a minute. We've already seen that calcium is stored in the endoplasmic reticulum. Now, one way to induce release of calcium from the endoplasmic reticulum store is actually to have an increase in the intracellular calcium concentration. Because you see, calcium binds to ryanodine receptors on the endoplasmic reticulum to activate calcium-induced calcium release. This is a feed-forward mechanism whereby an increase in intracellular calcium concentration can be amplified to produce a greater calcium signal. So why don't we actually call them calcium receptors? Well, like so many other receptors, they were named for the drug that was used to first discover and characterize them. The drug ryanodine is an antagonist at ryanodine receptors, so it blocks the ability of calcium to produce further amplification of calcium in the intracellular fluid. So Raymond and Redman found that ryanodine blocked LTP1. They also tested ryanodine in LTP2 and 3, shown in the bar graphs over on the right. The first bar graph shows that ryanodine knocked the strength of potentiation for one train of TBS stimulation down to about 10% of the potentiation in controls that were also administered one train of theta burst stimulation. And it shows that ryanodine did not disrupt the strength of potentiation when the sections were treated with 4 or 8 trains of stimulation, that is LTP2 or LTP3. Now the second bar graph shows that ryanodine decreased the time constant. Again, that is the measure of the duration of the potentiation to about 40% of controls for the sections that received only one train of theta burst stimulation. But it did not affect the time constant for the slices stimulated with 4 or 8 trains of stimulation. Accordingly, it seems that the weakest form of potentiation, LTP1, is dependent upon calcium-induced calcium release through ryanodine receptors. It seems that the calcium influx through NMDA receptors is not enough to sustain that initial period of responsiveness. It also needs the calcium-induced calcium release through ryanodine receptors. However, LTP2 and LTP3 are not dependent upon this mechanism. Hypothetically, they're dependent upon one or more other sources of calcium. Let's see if we can figure out what those sources are. When Raymond and Redman investigated LTP2 with 4 trains of theta burst stimulation, they challenged it with a drug called zestospongin. Now, zestospongin blocks the actions of inositol triphosphate, or IP3, at IP3 receptors on the endoplasmic reticulum. We saw IP3 receptors first in aplesia, and we've also seen evidence that IP3 signaling contributes to synaptic potentiation in LTP preparations. In this case, zestospongin decreased the potentiation for slices stimulated with 4 trains of theta burst stimulation, and decreased the magnitude and the duration, or time constant, of potentiation for LTP4. Once again, the effect was selective for only one stimulation protocol, that being LTP2. In the sections treated with LTP1 or LTP3, we saw normal magnitude and normal time constants, even when treated with zestospongin. So in this case, we can assume that the sections stimulated with one train of stimulation are able to experience the standard calcium-induced calcium release through ryanodine receptors. So zestospongin treatment really doesn't affect them. The control slices treated with 4 trains of stimulation open up IP3 receptors, and the effect of having those receptors open is far greater than the effect mediated by ryanodine receptors on the previous slide. But if the IP3 receptors are blocked, perhaps all these sections have to go on as the calcium-induced calcium release. So that's a smaller effect. And of course, the sections treated with 8 trains of stimulation must recruit additional mechanisms, so interfering with IP3 receptors doesn't appear to have a significant impact on them. Next, Raymond and Redman challenged LTP3, or 8 trains of stimulation, with the voltage-gated calcium channel antagonist, nifedipine. In these strongly stimulated slices, nifedipine decreased the potentiation for slices stimulated with 8 trains of theta burst stimulation, shown in the graph on the left, and decreased the magnitude and the time constant, or duration, of the potentiation for LTP3, shown in the bar graphs on the right. So once again, the effect was selective. Nifedipine, even though it had a substantial and statistically significant impact on LTP3, did not alter the magnitude or duration of potentiation for LTP1 or LTP2. Clearly, the voltage-gated calcium channels require more intense stimulation to activate them. Neither 1 nor 4 trains of stimulation can activate them, so nifedipine does not affect the impacts of 1 or 4 trains of stimulation. In the case of LTP1, the sections were only dependent upon calcium-induced calcium release. So nifedipine blockade of voltage-gated calcium channels that are not activated by LTP1 are irrelevant. And the same general situation is true for LTP2. Those sections are dependent upon calcium release through inositol triphosphate receptors, IP3 receptors, on the endoplasmic reticulum. And again, blockade of voltage-gated calcium channels is irrelevant. However, if the sections are activated by 8 trains of stimulation, the calcium influx through the voltage-gated calcium channels is much greater than the release from the intracellular stores in the other cases. So unless these sections are antagonized by nifedipine, the impact of activating the calcium channels is large, and it obscures the effects of ryanodine or zestospongin that were shown in the previous slides. So I'm going to sum this up by proposing a model to explain why there are different thresholds for activation of ryanodine receptors, inositol triphosphate receptors, and voltage-gated calcium channels. It's simply that one train activates ryanodine receptors, 4 trains activates ryanodine and inositol triphosphate receptors, and 8 trains activates ryanodine and inositol triphosphate receptors, as well as voltage-gated calcium channels. Ryanodine receptors must have higher affinity for calcium that activates them, so one train of TBS, of theta burst stimulation, is adequate. Then the inositol triphosphate signaling mechanisms have lower affinity for calcium, so it takes more trains of stimulation to activate them. And finally, the voltage-gated calcium channels have a high threshold for activation, so it takes a significant amount of stimulation, about 8 trains of theta burst stimulation, to activate them. Each of these successive forms of signaling drives more and more calcium into the cytoplasm, so the effect activated by more stimulation is not dependent upon the mechanism that was activated by the less intense stimulation parameters. The summary I described on the previous slide is actually supported by the fact that ryanodine receptors are prominent in spines directly where the stimulation is applied, keeping them close to the NMDA receptors where calcium influx occurs. Inositol triphosphate receptors are prominent in dendrites, so it may take a greater stimulation to activate them, as they're a short distance away from the site of stimulation. And we also know that we have to activate metabotropic signals. The voltage-gated calcium channels are especially prominent in the soma. So again, you should be able to see that it would take a more intense stimulation to activate these more distant mechanisms. So in 2006, Raymond and Redman followed up their original report by investigating the actual release of calcium using 2-photon microscopy with calcium imaging. In this very compelling study, they found that LTP1 released calcium right there in the spine where the ryanodine receptors are. LTP2 caused calcium release in the dendrite, but not the soma. And LTP3 caused calcium influx predominantly at the soma, where the preponderance of voltage-gated calcium channels reside. Now, the illustrations on this slide actually show calcium imaging through a dendritic shaft and a spine at the left, and a computer-generated 3-dimensional reconstruction of the endoplasmic reticulum extending up into a spine over at the right. So Raymond and Redman proposed a summary of the actions that occur in LTP1, LTP2, and LTP3. And that summary is depicted in the diagram at the right of this slide. They propose that LTP1, or one train of theta burst stimulation, activates NMDA receptors and allows a modest calcium influx into the spines, thereby activating calcium-induced calcium release, or CICR, through ryanodine receptors to activate kinases in the postsynaptic density. Then LTP2, which of course is 4 trains of theta burst stimulation, activates mGluR1 receptors to allow moderate calcium release from stores by activation of inositol triphosphate receptors both in the spine and in the dendrite. They propose that this, along with protein kinase C, activates local dendritic protein synthesis. And then LTP3, consisting of course of 8 trains of theta burst stimulation, activates L-type voltage-gated calcium channels. This allows significant calcium influx into the soma, which they propose triggers gene transcription. So in summary, this describes all of the mechanisms that are produced by weak and a little bit stronger and a little bit stronger theta burst stimulation. Now, did you notice in the last slide that Raymond and Redman invoked the activity of mGluR1 receptors in their explanation of the effects of LTP2, and even proposed that these receptors induced local protein synthesis? Did you think I was just sneaking those claims in there on the sly? Well, no. There's actually good reason to agree with these hypotheses. You see, in another set of experiments, Clark-Raymond reported that weak stimulation of Schaefer collateral inputs to CA1 pyramidal cells produces the expected small and decaying form of LTP that we've seen before. And we can see it here in the open circles in the top graph on this slide. But if he simply added the mGluR1 agonist, dihydroxyphenylglycine, or DHPG, the potentiation endured. So then Raymond challenged the mechanism of the mGluR effect, reasoning that the prolonged duration of the potentiation must be mediated either by activating local protein synthesis or by activating genomic mechanisms of protein synthesis. Accordingly, he challenged the mGluR receptor-mediated effect with the translational inhibitors cyclohexamide and emetine, or the transcriptional inhibitor actinomycin. So let's follow the logic here. If the effect of DHPG was due to mGluR1 induction of genomic transcription, then all 3 drugs, actinomycin, which blocks transcription specifically, and cyclohexamide and emetine, which block translation, all 3 drugs should block the effect of the mGluR receptors. On the other hand, if the effect of DHPG was due to mGluR1 induction only of local existing mRNA translation, then actinomycin should have no effect, but the protein synthesis inhibitors, the translational inhibitors, emetine and cyclohexamide, should each block the effect. Actinomycin wouldn't work because at the level of local protein synthesis, there's no transcription going on, and actinomycin blocks transcription. But the translation that's happening there would be blocked by emetine and cyclohexamide. So actually you can see in the middle graph that cyclohexamide and emetine, which is kind of in the foreground here, covering up the cyclohexamide effect, but they each blocked late LTP. And you can see on the bottom graph that actinomycin had no effect. And so this suggests that the mGluR effect, the effect of activating mGluR1 receptors, was to induce local protein translation, not genomic signaling. The text and diagram on this slide summarize the effects I've described. In brief, three mechanisms mediate the differing durations of LTP. We've learned from LTP1 that NMDA receptor-mediated calcium influx and ryanodine receptor-mediated release of intracellular calcium cause local spine increases in kinase activities that initiate LTP. And we've learned from LTP2 that metabotropic glutamate receptor activation of inositol triphosphate receptors causes dendritic release of intracellular calcium, enhancing local protein synthesis. And then we've learned from LTP3 that calcium influx through somatic voltage-gated calcium channels is due to action potentials, as we saw in the soma to nucleus model of genomic signaling, and this causes enduring LTP. And it seems that that occurs because of increases in genomic signaling. And if you consider this information all together, you should be able to see the calcium influx through the NMDA receptor is necessary for LTP. But this is not sufficient to produce enduring LTP, as the calcium effect must be amplified by release of calcium from endoplasmic reticulum intracellular stores and by calcium influx from the extracellular space. Okay, let's have a little fun, because we're going to wrap up this chapter by examining one of my all-time favorite demonstrations of the role of calcium in LTP. So strap yourself in, because this experiment is just really a pièce de résistance, published in Nature by Nicholas Tony and collaborators in 1999. And I suppose my French exclamation is appropriate, as Tony and the group worked at the universities of Geneva and Bern in Switzerland. Now, in this set of experiments, Tony and collaborators applied theta burst stimulation to Schaefer collateral inputs to CA1 pyramidal cells, as we've seen so often before. But they did something very smart, and I love smart experiments. They described their technique as potassium chromium trioxalate precipitation during osmium fixation. Okay, that's a mouthful. So would you like me to translate? Sure. And yes, I seem to have got into a habit of talking to myself. Oh my. Okay, so here's the story. The potassium chromium trioxalate precipitation during osmium fixation method really simply means that the experimenters stimulated the hippocampal section. Then they fixed the sections, or processed them so that all proteins are cross-linked and held in place, maintaining the tissue in pretty much exactly the state it was at the moment of fixation. The special thing about their fixation technique is that it precipitates the calcium into clumps. So you can see the dark masses in the spines if there was calcium hanging around inside the spine. They stimulated the sections with theta burst stimulation and then fixed the tissue 5, 15, 30, 45, 60, or 120 minutes later. However, in every case, they also stimulated the tissue 5 minutes before fixation. Accordingly, the tissue processed 5 minutes after stimulation got one set of theta burst stimulation. The others got stimulation and then a second stimulation 5 minutes before fixation. An important point is that the tissue section was left completely undisturbed between stimulation 1 and stimulation 2. So the first stimulation would set into motion any forms of modification that would occur in the spines. The second stimulation would activate exactly the same spines again, and in those specific spines would incur release of calcium from the endoplasmic reticulum. If they then precipitated the calcium in the tissue, the calcium markers indicate which spines were affected by the first round of theta burst stimulation. Okay, have you got it? The first stimulation activates some but not all the spines in the slice. During the interval of, well, 15, 30, 45, 60, or 120 minutes after the stimulation, that calcium signal would be cleaned up. It would be gone. However, they relabeled the same spines with a second round of theta burst stimulation and were then able to visually identify which spines in the section had been originally potentiated. What they were actually looking for was visible changes in the structure of the spines through the electron microscope. In the image on the slide, you can see a red circle around a calcium precipitate with a black arrowhead pointing at it. Inside the blue circle is another dendritic spine that clearly shows no evidence of a calcium precipitate. So by comparing the labeled and unlabeled spines, they're able to see any differences between the spines that were activated and those that were not. They were also able to examine the impacts of drugs that would be expected to block potentiation-related changes in order to validate their approach. In the upper graph, you can see that about 5% of the spines were labeled in sections that did not receive theta burst stimulation. Similarly, about 5% of spines were labeled with calcium precipitate if they were treated with APV and then stimulated. Of course, the APV would block the NMDA receptors, so we would expect no calcium influx and none of the downstream effects. So about 5% is the baseline. This is how many spines are labeled in a section that has no theta burst stimulation, probably an artifact, the 5% being an artifact of sectioning the tissue and preserving it in a bath. But moving farther along the x-axis on this graph, we can see that sections stimulated and then processed after 30 or 60 minutes, with that second round of stimulation, of course, exhibited more than a doubling of the numbers of spines with calcium precipitate. Also notice that the last column on the graph shows that treatment with KN93 did not affect the number of potentiated spines. KN93 is a kinase inhibitor, so it may impair the changes in the spine as it's downstream from the actual calcium influx, but indeed it did not affect the number of labeled spines. The second graph shows that the density of the precipitates at 60 minutes, measured as the area of precipitate over the spine profile area, varies between spines that were stimulated. And then the bottom graph shows the strong potentiation they got with their theta burst protocol and the impact of KN93 on the potentiation, really just documenting that this is a normal preparation with calcium-dependent kinase activation underlying the potentiation of the synapses. Now, the principal reason they did this experiment was that neuroscientists had repeatedly reported a phenomenon called perforated synapses. They showed up rarely in these kinds of ultrastructural analyses under the electron microscope, but they were reliably observed. Nobody knew for certain what they did, but some scientists had proposed that they were a mechanism of increased synaptic strength. The concept is illustrated in the figure at the lower left. By actually splitting the synapse, the terminal would now have two active zones on the postsynaptic spine. But nobody had figured out how to test if these rare profiles were really even associated with LTP, never mind a stable form of synaptic potentiation, well, until Nicholas Tony and colleagues came along. Let's see what their evidence tells us about the potential role of perforated synapses in LTP. But a little warning, it may not be quite what you expect. The photomicrograph at the upper right shows a perforated synapse. The axon terminal is at the right side of this photomicrograph, and the spine is over at the left. I can tell because the structure at the right side of the picture is filled with neurotransmitter vesicles, as an axon terminal should be. Note also that this axon terminal and spine represent a stimulated input, as the small arrowhead points at a calcium precipitate. And then note especially the two distinct postsynaptic densities, there and there, with the single axon terminal. The large arrowhead points at a perforation in the postsynaptic density, or a gap between the two densities that are now evident in the perforated spine. This is the definition of a perforated synapse, and this is a superb photomicrograph to illustrate it. So if you were a proponent of the school that proposed that perforated synapses formulated a basis for the permanent changes in synaptic structure that underlie synaptic potentiation, your expectation would be that perforated synapses increase after potentiation, and that they endure, creating those permanent changes in spine structure that underlie potentiation. And that made great sense. The axon terminal is still talking to the same postsynaptic dendrite, but instead of one active zone, it's talking to two active zones, thereby maintaining the accuracy of the information being conveyed, but doubling the input. However, let's take a look at the graph. It shows the mean number of synapses per labeled spine in the slices across time. We can see that the expression of perforated synapses increased across time, going up to nearly 50% of potentiated or labeled spines by 30 minutes. But thereafter, the numbers of perforated synapses actually declined, coming back to near initial levels by 60 minutes. So, it seems that perforated synapses do not provide the permanence to the potentiation that some scholars had proposed. Of course, there were no changes across time in perforated synapses in unlabeled spines, remaining at around 5%. But these experimenters found an additional interesting change when they examined the labeled activated spines. They found the multiple spine profiles that are shown in the middle photomicrograph. Again, the spines are on the left and the axon terminal is on the right. In this case, you can see a beautiful rendering of a single axon terminal that is in contact with two mature and functional dendritic spines that are labeled with calcium precipitate. So let's look at the graph underneath this photomicrograph. This graph actually shows that the number of multiple spine profiles increases from less than 10% of labeled spines in the early part of the time course to stabilize at more than 50%, at more than 15% of the activated spines. This indeed seems to contribute greatly to the strengthening of responses to presynaptic input in the postsynaptic neuron. And this seems to be the permanent change that endures across time from 60 all the way through to 120 minutes. Overall, the research team identified that perforated synapses peaked at 30 minutes and multiple spine profiles peaked at 60 minutes. However, the perforated spines declined after 30 minutes, whereas the multiple spine profiles persisted beyond the peak. What seems to be happening is that the perforated synapses, shown again at the left on this slide, seem to be an intermediate phase, so that the potentiation of the synapse causes this initial perforation in the postsynaptic density in that spine. But then as time goes on, that progresses to an actual splitting of the spine structure to yield two mature and functional spines on that axon terminal. Now, importantly, the elevations in perforated synapses and in multiple spine profiles were not observed in sections treated with the kinase inhibitor, shown in the bar graphs on this slide. The KN93 treated sections were labeled with calcium precipitate, but there was no increase in either of these morphological profiles, as you might expect, if the changes were associated with calcium-dependent activation of kinase actions. And this is way cool. So finally, when Tony and collaborators examined the new multiple spine profiles, they found that the majority of them arose from the same dendrite. This is shown brilliantly in the photomicrograph at the bottom of this slide. This is just such a perfectly aligned profile. Imagine they're sectioning tissue embedded in paraffin for electron microscopy. In other words, they're slicing the tissue blindly. They cannot in any way see the submicroscopic structure when they're actually sectioning the tissue. So to get a section that is so perfectly aligned to show the length of dendrite and two perfectly aligned spines here and here is simply stunning. They must have had a party when they examined the tissue under the electron microscope and recorded such a beautiful picture. But now for the rest of the analyses, they would have had to painstakingly examine serial sections, and they would have had to use computer-generated algorithms to formulate the kind of three-dimensional reconstructions that are shown in the upper images. The image at the top right shows a multiple spine profile on an axon terminal where the spines originate from distinct dendrites. The lower two computer-generated images result from spines that originate on the same dendrite. So the graph and the data reveal that in labeled spines that were activated by the theta burst stimulation, 60% of the multiple spine profiles originated from the same dendrite, lending great credence to the mechanism described in the previous slides, where a single postsynaptic density was perforated and then the spine was split and formed two distinct synapses with two mature spines. And I'll wrap up my commentary on these experiments by saying, alors ces expériences sont incroyables, or wow, these experiments are just incredible. However, there's one remaining comment that I must make. In the conclusion of Tony's Nature paper, they wrote, together, these results indicate that the late phase of LTP is likely to be associated with the formation of new and possibly functional synapses between an activated terminal and its target cell. This mechanism of synapse duplication maintains specificity in information processing and provides a framework for understanding the numerous in vivo experiments that have demonstrated an increase in synapse number and complexity of dendritic arborization following exposure to an enriched environment or learning paradigms. Now, there's nothing specifically incorrect about this statement. However, I feel like it misses part of the bigger picture, or it just doesn't address that bigger picture. They seem to want to attribute learning and the impacts of enriched environments to formation of multiple spine profiles. However, the problem with this conclusion is that environmental enrichment and some forms of learning appear to result in increases in dendritic arborization, not just new spines on single dendrites. And this is illustrated in the figure on the left. The jewelfish, of all things, reared in an enriched environment, and believe me, I have no idea what an enriched environment is for a fish. But the jewelfish reared in an enriched environment has more elaborate dendritic architecture with more spines on more complex, extensively branched dendrites. And furthermore, not all learning consists of elaboration or expansion of dendritic structures. Pruning can also occur, and this is shown in the case of the myna bird on the right. Now, the juvenile myna bird has a massive density of spines on dendrites of neurons in the song region of the brain. And then after learning the species-specific song, the spine density is extensively pruned to leave a much less dense neuropil. In other words, not all learning is instructive, meaning not all learning consists of elaboration of dendrites and spines. Some learning is actually selective, choosing from among many alternate structures to refine the correct set of mechanisms to underlie the formation of the internal representation. Now, neuroscientists in general have paid much less attention to this latter form for encoding of memories.